Microbes are in us, on us, and all around us. They have a wide range of functions. They can cause the decay of materials, ferment sugar to make wine, and produce antibiotics. But did you know the discoverer of microbes wasn't a scientist? He was a draper. Drapers used magnifying glasses to inspect the quality of their fabrics. Antony van Leeuwenhoek was a draper and merchant who, unhappy with those lenses, taught himself glassmaking. He created high-quality tiny glass spheres that could magnify objects up to 270 times. His microscopes were less than 2 inches long and were used by holding one's eye close to the tiny lens and looking at a sample suspended on a pin. As a good merchant, he knew to keep his microscope secret, but that wouldn't be possible for long. Van Leeuwenhoek was a curious man, so he started analysing anything he could get his hands on. He sent his discoveries on mould, lice and bees to the Royal Society in London, who were familiar with similar work. However, his next discovery caused the scientists to question his credibility, as it was something completely unknown to them. Van Leeuwenhoek had come upon an infinity of tiny animals in pond water, which he called animalcules. They are now known as microbes. This was the first ever sighting of single-celled organisms, so no wonder the Royal Society didn't trust him. Van Leeuwenhoek was forced to show his microscopes to various political and religious personalities they sent to prove he was of sound mind, and they finally believed him. Van Leeuwenhoek never wrote a book. His discoveries were all communicated through hundreds of letters to the Royal Society. These included the description of blood cells, the confirmation that blood travels through capillaries, and the sighting of moving cells in human sperm, which helped refute the spontaneous generation theory. Science would never be seen with the same eyes again.
Everything that lives is made of cells, even us. Although we can't see them, trillions of cells within our body work together to give us the gift of life. These tiny units of life fascinate biologists like Boyce Rensberger.
The entire human body is made out of cells or the things that cells have made, such as bone. Skin is made of cells, your heart is made of cells, the muscles that operate the body so that you can move, these things are all made of cells, including the brain itself, which sends electrical signals around from one cell to the other so that we can think. All these different kinds of cells work fundamentally the same way. When you look inside the cell, you see the same kinds of structures.
Cells come in all sorts of sizes and shapes. They're so small that we need a microscope to see them. Inside of every eukaryotic cell is a microscopic factory that runs off chemical reactions. At the center, the nucleus acts as the cell's brain and controls its activities. It also holds the DNA, the instructions for life. The cell's membrane protects each cell and takes in the raw materials it needs to stay alive. Water, nutrients, and waste move into and out of the cell all the time. These materials may hitch a ride along the cell's transport network, a network made up of microtubules and intracellular membranes. The cell's transport network picks up, transports, and delivers material throughout the cell. The cell breaks down raw materials into the building blocks it needs to grow. This ability to take in vital materials allows the cell to function and perform specific tasks.
The instructions that are in the cells, starting in embryonic development, tell each cell how to change to take on a new function. Also, the cells monitor the activities going on. If there's a wound, the cells can sense that, and they will send out the message to other cells to come and repair the damage. So all of this keeps the whole body on a kind of an even keel.
Our cells work together in extraordinary ways. The heart, for example, is made up of muscle cells with a tendency to twitch. Separately, they twitch to their own rhythm, but together they beat as one. Millions of them work together to give us a heartbeat. These muscle cells survive as long as we do, but throughout the body, cells die and get replaced all the time. Our body produces new cells through mitosis, the process where one cell divides into two. Since we're constantly losing and replacing cells, most cells in our body are actually younger than we are.
This is the historical background of the nature of cell. In 1665, British scientist Robert Hooke observed mass tiny cavities from thin slices of cork with his self-made microscope. He named these structures cells. These structures reminded him of the small room in the monastery. 1674, Dutch scientist Anton van Leeuwenhoek made pioneering discoveries concerning protozoa, red blood cells, capillary systems, and the life cycles of an insect. He also perfected the construction of the compound microscope. 1831, British botanist Robert Brown observed plant cells with a distinct central part and coined the term nucleus. He also described the streaming movement of the cytoplasm and called that the Brownian movement. 1835, French biologist Félix Dujardin observed that cells were not empty but filled with thick jelly-like fluids called the protoplasm. 1838, German botanist Matthias Leiden concluded that plants are composed of cells and formulated the plant cell theory. In 1839, German zoologist Theodor Schwann concluded that animals are composed of cells and formulated the animal cell theory. And in 1858, German pathologist Rudolf Virchow concluded that all cells must come only from pre-existing cells. 1932, German engineers Maxwell and Ernst Röskopf built the first transmission electron microscope. By 1953, James Watson, American biochemist, and Francis Crick, British biophysicist, discovered the structure of DNA that ushered in the era of molecular biology.
The microscopes used today are far more complex than those used in the 1600s by Anthony van Leeuwenhoek, a Dutch shopkeeper who had a great skill in crafting lenses. Despite the limitation of his now ancient lenses, van Leeuwenhoek observed the movements of protista, a type of a single-celled organism, and sperm, which he collectively termed animalcules. In 1665, publication called Micrographia, experimental scientist Robert Hooke coined the term cell from the box-like structures he observed when viewing cord tissue through a lens. In the 1670s, van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses, microscope construction, and staining techniques enabled other scientists to see some components inside the cells. By the late 1830s, botanist Matthias Leiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory. The unified cell theory states that all living things are composed of one or more cells. The cell is the basic unit of life, and new cells arise from existing cells. Rudolf Virchow later made important contributions to this theory. Leiden and Schwann proposed spontaneous generation as the method for cell origination, but spontaneous generation, also called abiogenesis, was later disproven. Rudolf Virchow famously stated, "Omni cellula e cellula." All cells only arise from pre-existing cells. The parts of the theory that did not have to do with the origin of cells, however, held up to scientific scrutiny and are widely agreed upon by the scientific community today. Cell theory was proposed by the German scientists Theodor Schwann, Matthias Leiden, and Rudolf Virchow. The cell theory states that all living species on Earth are composed of cells. A cell is the basic unit of life. All cells arise from pre-existing cells. A modern version of the cell theory was eventually formulated, and it contains the following postulates. Energy flows within the cells. Genetic information is passed on from one cell to the other. The chemical composition of all the cells is the same.